Morning, folks. Thank you, as always, for being here early. Morning, Anna. Hello, those in 10 Evans. I forget who it is that said that problems facing humanity are parsable into two types. There are puzzles and then there are mysteries. A mystery is something where we don't even yet know how to begin to answer the question. A puzzle is one where we don't know the answer, but we know how to begin to address the question. Heredity has been observable since the beginning of consciousness, so I suppose since Homo sapiens and the chimp diverged about 1 million years ago. But it wasn't until literally the past 100 years that the concept of heredity went from being a mystery, as in we don't know how to begin to think about it scientifically, to being just a puzzle. Well, not just, it's still quite substantive, as in we can start to pose questions that we know how to try to find answers to. I taught you that the remarkable diversity of coat in purebred dogs, the Airedale Terrier or the Basset Hound, is explicable in terms of variation at only 3 gene loci. 1 dominant, 1 recessive allele at each one. And then when you mix and match them like some sort of genetic molecular Lego, you get this marvelous diversity of phenotypes from only 3 genes and only 2 alleles at each gene. I also taught you that the pristine uniformity of appearance of purebred dogs is parsable into a kaleidoscope of phenotypic diversity through only 2 rounds of breeding and 2 rounds of meiosis. The first generation all looks like grandpa, and then when grandpa makes gametes and grandpa's sister grandma also makes gametes, meiosis just moves alleles around and forms these new combinations, and when the gametes fuse, you get this remarkable diversity of appearance. This, of course, is useful because it can explain why diversity can be generated through sexual reproduction, and of course is highly useful in forensics. I told you that when meiosis moves alleles around, alleles of CODIS loci, short tandem repeats that live on different chromosomes, segregate independently of one another. And as a consequence, we can use the product rule to ask whether, let's say, the suspect's DNA matches the DNA found at the crime scene. So I am hopeful that by now, after having spoken about this for about an hour non-stop, I have convinced you that meiosis shuffles alleles. Some of you may have noticed that I haven't yet explained why I'm teaching you this. I'm teaching you this because we have before us a puzzle. Not a puzzle, but a mystery. Sorry, not a mystery, not something where we don't know how to begin, but a puzzle. And it is shown here. This is a figure from your textbook. And it represents one of the most genetic of all human traits. And that trait is height. As I'm about to explain to you, human height is highly heritable. Now, the first thing for you to learn for the rest of your life, that highly heritable doesn't mean unchangeable. So our ancestors, our great, were significantly shorter than us. That had not to do with the fact that they had shorter genes. They simply lived in a different environment. That said, height is highly heritable, and I can walk you briefly through what is known about this before posing the puzzle to you. This is a visual representation of what happens to mankind, humankind, if you parse it by height into a histogram. You get this. Any math minors, what is the mathematical equation that explains this? Somebody raise their hand and say it. Sir? The normal curve. What's the equation? I know it's early. Sir?
A sine function?
No, not a sine function. E to the minus x squared. It's the bell curve, the Gaussian curve. E to the minus x squared. The standard way to refer to this bell-shaped distribution, believe it or not, is, quote, the normal distribution. So when geneticists talk about the fact that a given trait, such as height, is normally distributed, that word, that adjective, has a technical meaning. It means it follows the Gaussian curve. So most people are of average height. There are some extremely tall people and some extremely short people. Over the past 7 years, due to an extraordinary effort in this regard by geneticists all over the world, we have identified the loci in the genome that cause height to be highly heritable. So the fact that the offspring of two taller than average people will tend to be taller than average, if grown under the same environment, of course, and if that the offspring of two people shorter than average will tend to be shorter than average as well, again, if grown under the same environment. Before I show you the data, I need to give you a theoretical example, but I promise you, I will show you a picture of Michael Jordan right after that, so please stay awake. This gentleman, who, to be frank, looks slightly mentally unstable, is in fact the great geneticist Herman Nilsson Ehle, and it's completely cheap to take shots at people's photographs from a century ago. He simply wanted to ask, do we have to have binary phenotypic differences, yellow versus round, green versus wrinkled? I'm joking, of course, yellow versus green, round versus wrinkled. As I'll teach you today about Drosophila, red versus white. He realized that, of course, in nature, very few things are that binary. For example, there isn't tall versus short. There's slightly taller versus slightly shorter. It turned out, as he discovered theoretically and was then shown experimentally, Mendelian processes are in fact perfectly compatible with having a continuum of phenotypes. Let me give you an example. We have to imagine a trait, this is a hypothetical trait, I'll show you a real-world example in a second, controlled by only 3 loci, A, B, and C, each of which occurs in only 2 alleles, big and small, dominant and recessive. Here is the core intellectual trick. You have to imagine that the effects of these alleles is somewhat additive. In other words, an all homozygous recessive organism is all white, whereas an all homozygous dominant organism is dark red. However, even one extra dominant allele changes the color a little bit. So you cross a homozygous recessive square to a homozygous dominant square, you get an intermediate 3-locus heterozygous square, which is intermediate in color. And when this peculiar kite makes gametes, of course, Mendel's second law starts waving its flag all the way from Brno. We get all of these gametes, both male and female. When they fertilize each other, we get the exact same distribution I showed you in dogs last time. But remember, we made the assumption that the effects of alleles are additive. So even one additional dominant allele will cause a small change in color. If we then rank the resulting entities all the way from the one that just looks like the dark red kite, grandma, and all the way at the other extreme, extremely pale white, grandpa, we will notice that we get 1, 2, 3, 4, 5, 6 gradations of color from one extreme to the other, and that they occur in the following numbers. So folks, in just 1 to 2 generations of breeding, from a simple Mendelian process, we've gotten ourselves a Gaussian curve. Telling you this because of this. This, of course, is Michael. And now, I think Michael is 6'8", 6'9", does anybody know? 6'6". Michael is 6'6". And this is a very accomplished point guard. His name is Muggsy Bogues. He was one of the shortest people to play at that level professionally in the NBA, and I think he was 5'10". So he was... 5'3"? Okay, so he was 5'3". He was quite good. No, he was really, really good. He was a very accomplished player. It's just that, the standard, Michael is pretty tall, and he was not particularly tall by NBA standards. If you look at the situation as of March 2012, not at the one in the NBA, but the one in human genomics, you will learn that the heritability of height, it's a technical term that means how much of the difference between individuals is due to the additive effect of genes. What does additive effect of genes means? It means this type of process. An additional allele changes the phenotype by how much? 80% of the variation for this trait is due to variation in their genes. That's a lot, isn't it? 80%. So in order to find where those genes are in the human genome, scientists all over the world actually looked at the height and the DNA of 180,000 people. That's not a small number. And they identified, believe it or not, 180 different loci in the human genome that affect height. Woohoo! Well, no, not really. Remember I told you that 80% of the difference between humans is explained by genetics? The alleles they found explain only 10% of the variation. And they looked very hard in a very large sample. Each tall allele, in other words, by analogy with this, each tall allele of those 180 genes that control height changes height by how much? By 0.3 centimeters. So, percentage of samples studied, number of tall alleles, approximate height difference in centimeters from 0 to 1. So if you get one extra taller allele, you're taller by that. So we have to conclude from these data that the difference between Michael and Muggsy is explainable by a very, very large number of loci. And you will say to me, well, it's because they didn't try hard enough. Folks, let me show you what the author list looks like on that publication. This was a big study. So before I dive into science, I thought I'd show you something silly briefly, just to wake you up. I never thought in my entire pedagogical career I would ever show my students anything from the website glamour.com. But I did want to connect what I just showed you about the heritability of human height to what I showed you in the single and then second generation segregation of pedigrees and phenotypes for multiple locus inheritance. So this is what you get. This is the distribution of phenotypes you get with only 3 loci, 2 alleles each. Human height is controlled by hundreds of loci and potentially multiple alleles each. So let's think about meiotic dynamics and the number of different gametes that a human can produce. When multiple hundreds of loci start playing Mendel's second law game, Mendel's second law, the game, with 23 separate chromosomes. So again, I never thought I would go to this website, but here it is. I went to glamour.com, and if you conceptualize a union between these two individuals, as this gentleman and this lady choose to have a child, that child, of course, will be heterozygous for the hundreds and hundreds of loci in his or her genome, one from tall mom and one from not tall dad. And of course, then this will play itself out. But when you play with 2 alleles for 3 loci, you get 8 different kinds of gametes. If you play with 600 loci and multiple alleles each, can you imagine what meiosis will do to the F2? Okay, and now the serious part. I taught you that in peas, single alleles of single genes have enormous effects on phenotype, in, round versus wrinkled. In humans, that also happens, but such effects are rare. So for example, BRCA1 or 2 mutation-induced breast cancer accounts for 5% of all cases. Now, this doesn't mean it's not tragic or important. Of course, it's both. It's just rare. 95% of human breast cancer cases are not explicable in simple Mendelian terms. Now, in dogs that have been the subjects of extensive artificial selection, breeders, the Victorian breeders that had a lot of spare time on their hands, pulled out alleles with very strong effects. Remember the pug and the Saint Bernard I showed you at the beginning of last class? That's one allele. That's a very strong allele of the IGF-1 gene. In humans, such alleles exist, but they are rare. Similarly, I showed you that the Sprint Alaskan dog is highly heat tolerant as compared to its ancestor, the conventional Alaskan sled dog, due to an allele of myosin. Again, such alleles in the human population are rare. Humans haven't been artificially selected. That's just a fact. And so differences in our traits of interest, as best as we can tell, whether it be height or perhaps more importantly, cardiovascular disease susceptibility, which we know has a genetic component, or susceptibility to diabetes, both 1 and 2, which we know has a genetic component. appear to emerge from the combined effects of variation at hundreds and hundreds of genes. So this makes, frankly, for a very interesting challenge moving ahead for my generation, of course, but for your generation even more significantly. Because objectively speaking, the way to move forward is not to throw your hands up on there and say, oh, there are hundreds of loci. What are we going to do? We have to move forward. We cannot stand still. So we need to understand how traits and differences in them come about from the action of genes. How does height emerge out of the action of the human genome? How does cardiovascular disease emerge out of the action of the human genome? And that, of course, is what we're going to look at next. How do we compare genes to traits from a real organism perspective? I want to also emphasize that this topic, to me as an American educator, is particularly important because, of course, the US of A is the world's greatest melting pot. As I've spoken to you about this briefly when I talked about the genetics of immunological identity and the challenges of finding bone marrow donors or other organ donors for individuals of mixed race. Here in the US, we have this extraordinary constellation of individuals of unique histories, whether you have a businessman and a software programmer who's Syrian American, or an athlete who is Asian African American, or a musician who is African American, or a newscaster who is Japanese American, or a president who is Kenyan American. Not only is it fascinating from a sort of glory of human diversity perspective, this makes for some really interesting meiotic dynamics. And I realize that what most people look at Alicia Keys or Ann Curry, they don't think about meiotic dynamics. But scientists do. They go, oh, segregation and pedigrees, oh. And I will actually talk to you a little bit about this as I walk you through genes and chromosomes. I'm very sad to say that the person in this picture who is in your textbook is doomed to die by age 40. And this is because this person has cystic fibrosis. It's the most, probably the most common autosomal recessive disease in the Caucasian population, and I use that word on purpose because I'm going to get to that in just a second. Its prevalence estimate is 1 in 2,500 live births. Now, 78 is the life expectancy of an average person in the United States today. Individuals with CF, this is what's called a Kaplan-Meier curve. Kaplan-Meier. It basically shows you that by age 40, whatever the country and whatever the standard of care, individuals with cystic fibrosis are unfortunately doomed to die. What are the primary symptoms of people with CF? Their lungs clog up with mucus. They get pulmonary infections, and that leads to respiratory failure, and their pancreas fails. Treatment for cystic fibrosis patients until relatively recently, I mean literally as recently as a year ago, was largely palliative. This means that we could try to control the symptoms, but couldn't do much to affect the root cause. This offers me an opportunity to teach you multiple useful concepts as we transition from the era of classical genetics to the era of molecular genetics, so sort of 1953 onwards. And I'm going to begin by acknowledging the man whose work led to the identification of the gene mutations in which cause cystic fibrosis. It's Dr. Francis Collins, and he happens to be the director of the National Institutes of Health. His lab found that the gene mutations in which cause cystic fibrosis lies on the long arm of chromosome 7. The gene is called CFTR. You don't need to know this. Cystic fibrosis transmembrane conductance regulator. It's a channel. It allows for ions to pass through cell membranes. This is slang that you will learn, I suppose, next Monday. This is genetic nomenclature for the kinds of mutations that have been found in individuals like this person, somebody with CF. A deletion of an entire codon, so a deletion of 3 base pairs. A nonsense mutation, so a change in the genetic code which causes the translation of this messenger RNA to stop prematurely. A missense mutation in which one amino acid is replaced by another, and so on and so on. The teachable moment that will thus reoccur from now until the end of the section is that different mutations in the same gene, so strictly speaking, different alleles, allelos is the Greek for different, can yield the same phenotype. What are all of these alleles doing? They're inactivating the function of the gene product. Why do they lead to the same phenotype? Well, the teachable moment here is the gene, of course, as initially identified by Mendel, is the unit of biological function. Now, I showed you a picture of Ann Curry, who's Japanese-American, and Alicia Keys, who's African-American, and other folks like them, for an additional reason, not just to talk about the excitement of meiotic dynamics, which are exciting, but to talk about the challenge of genetic counseling. A challenge, I think, unique to the United States, but one that I think will spread through the world. Specifically, humanity's history is inscribed in its genome, and specific groups with specific ancestral history are known to have different alleles of different genes at different frequencies. Specifically, alleles of the CFTR gene that cause cystic fibrosis are apparently 4 times more common in non-Hispanic Caucasians than in Asian Americans. So a non-Hispanic Caucasian, the carrier frequency is 1 in 25. This means that the risk of being a carrier couple is 1 in 625, and the risk of having a child with cystic fibrosis is 1 in 2,500. If you're Asian American, then the risk drops. The teachable moment here, humanity's history is inscribed in its genome. Now, imagine a situation where an individual arrives in your office and says to you, Dr. Lee, am I at risk for CF? And you say, well, Mr. So-and-so, tell me about your family background. And then you get this classic, only in America, kaleidoscope patchwork quilt of ancestries, and you go, oh, so much for the ethnicity-specific frequencies then. In fact, the American College of Obstetrics and Gynecology, good for them, and that, by the way, is a hyperlink that you can go to read about the current guidelines. In recognition of the fact that America is a melting pot, has now recommended preconception carrier screening for cystic fibrosis for all individuals, irrespective of their self-reported ancestry. Because at this point in the USA, that seems to play less and less of a difference. Incidentally, how would you preconception carrier screen for cystic fibrosis? You don't have to do this if you don't want to, but this will be kind of fun. If you go to this website, the American College of Medical Genetics, you will learn, not surprisingly, that you do this using a technique that you learned here from Professor Meehan and in Professor Meehan's laboratory, the polymerase chain reaction. With that, here's a tiny, tiny homework. Discuss this with your GSI from start to finish. Imagine you are hired by a starting Bay Area startup who wants to market a quick diagnostic kit for cystic fibrosis, the mutation. So a couple shows up and says, Dr. Chen, we're Caucasian. We've heard that we're at a higher than average risk for being carriers for the cystic fibrosis calling allele of CFTR. What specifically would you do, like literally from start to finish? From what do you tell the patients to do? I beg your pardon, the individuals who visited your office to do. And by way of a specific example, this is the most common cystic fibrosis causing allele in non-Hispanic Caucasians. It's a deletion of 3 base pairs, this TTT, AAA, and it accompanied by a loss of 1 phenylalanine from the opening reading frame. So the answer to your homework should be a list. Step 1, panic. Step 2, pull out your notes from Professor Meehan's laboratory and PCR. Step 3, and so on and so on and so on. I really need to credit Francis Collins for not just having discovered the gene mutations which cause cystic fibrosis in his lab, but for having really pushed genomics, the study of the genome, to the forefront of the biomedical enterprise. Only 5 weeks ago, the Food and Drug Administration has taken a remarkable step, which followed an even more remarkable development in the pharmaceutical industry. Now, preconception and carrier screening for cystic fibrosis causing alleles of CFTR is all fine and dandy, But that has to do with future generations, children that are about to be conceived. Folks, there are 70,000 people with CF alive today, doomed to die by 40. They want to live to 78 and beyond. And so, in a remarkable validation of the fact that studying the human genome is worth it, a company called Vertex discovered, put through clinical trials, and showed the efficacy of a small molecule drug that fixes the defect. Believe it or not, it fixes the defect of that allele of CFTR. It's a molecular agent that goes in and fixes the mutant protein. And it is the first such targeted drug in the entire history of cystic fibrosis approved, and it does marvels for patients. On the Vertex trial, there were, believe it or not, I know this sounds hard to believe, this is true. There were two twins. I cannot show you their images because they're not public. And one of them was in the placebo arm, not on purpose, it just happened. And one was in the treatment group, and they walked out of the elevator to walk to the nurses for their checkup after taking the drug, and the nurse started to faint because it was so clear which one of them was taking the medicine. This is good stuff. Look it up. I am telling you this because Francis Collins identified the gene mutations in which cause cystic fibrosis via a technique known as mapping by linkage. So there is a direct path from this life-saving drug discovered by Vertex all the way back to 1913, a laboratory at Columbia, and two sad undergraduates who lived on the same food as their model organism. Does anybody know what that is? What food, what one model organism lives on food that humans can eat as well? Yeast. So you're suggesting we drink YPD? Bananas, Drosophila. Back then, Drosophila was grown on mashed bananas. It was perfect. It fed the hungry undergraduates and the fruit flies that worked on them. Mapping by linkage allows us to take a number of families where a condition of interest occurs. So here's a family, and we have a strong hunch that the occurrence of whatever condition that is indicated here by the filled circles and squares is genetic. We then analyze their genomic DNA and determine where in their genome, so on what chromosome and in what gene, the genetic cause of this condition lies. Now, as you shall see shortly, mapping by linkage involves these marvelously elaborate genetic tricks, crosses, maps, this, that, the other. Surely, one would argue, you simply sequence the entire genome of a person with the disease and the entire genome of the person without the disease. And you compare them and you say, aha, phenylalanine. There's only one minor problem, which is there are a good number for you to know. 6,000 coding differences between two randomly chosen people. This question has been studied experimentally. If you compare all the genes, all the coding sequences of everyone in the room, on average, 6,000 coding changes. And the only remaining question is which one of those causes the disease. Furthermore, are you kidding me? Sequence the entire human genome? Wouldn't that take like forever and a lot of money? Well, until as recently as 5 years ago, everyone would have laughed at you if somebody would have said, just sequence the genome and look. Until this happened. I still remember when this paper came out and trying to reattach my lower jaw. The scars are still there. This was work from Jay Shendur. He's a professor up at UW, and his grad student, Sarah Eng. And I swear, Hermione and her wand must have helped them, because there must have been a spell issued. Find me the mutant position. I cannot do justice in the time that I have to how this was done. This will not be on the midterm, but if you choose to go to graduate school or medical school or become a public health professional, pretty much anything other than trading in collateralized debt obligations, I encourage you to learn about this. It's remarkable how this was done. It was quixotic, but it worked. Having said that, however remarkable is the fact that you and I live in an age where it's feasible to sequence the entire genome and say, actio lossis mutandis. That is not the standard. It might become so by the time you graduate from grad school or medical school or public health school. So it is amply worth our time to understand how mapping by linkage occurs. To explain to you how mapping by linkage occurs, I need to introduce you to a model organism that you've already met, and I'm hopeful love dearly. It's amazing, isn't it? We've learned so much about us by not studying us. Yes, sure, there's nose te ipsum, know yourself. It should be nose, your model organism. For example, Strongylis trotus purpuratus taught us that the nucleus is where heredity lies. Batrachoceps attenuatus taught us that there are chromosomes, and chromosomes go through mitosis. Pisum sativum is not only yummy, but taught us that there are genes, and they come in forms known as alleles, and that alleles behave in really interesting ways during meiosis. Folks, I give you she who loves the vine and has a colored belly. Drosophila melanogaster. She taught us that genes lie on chromosomes and that they lie on chromosomes in a linear order. And we owe her existence and dominance of modern biological research to one man, the closest that the 20th century has to Gregor Mendel. Gregor Mendel's heir in the history of genetics, the founder of modern genetics, the first native-born American to have won the Nobel Prize, a pride of our nation, Thomas Hunt Morgan. He was a truly, truly remarkable man. And he moved the planets, scientifically speaking. After him, the world became different. And he did so using Drosophila. This was in Colombia in between 1905 and about 1940. He won the Nobel Prize for his work in about 1930-something. He was interested in the problem of heredity and chose the fruit fly due to some advice from a Berkeley entomology professor. Because it was easy to grow, it would live on mashed bananas, and the undergraduates who worked with it would also live on mashed bananas. And it was hardy. It did not require much. It bred fast. You could breed many of them. And it was an animal, so it wasn't a pea, so you could learn something about animal genetics. Morgan struggled mightily with Drosophila for the first couple of years because he would just breed these fruit flies, and all the offspring looked like the parent. And he, to his enormous credit, he persevered. And after a few years of breeding like with like to get like, to cite a book about this, May 1910 was when the revolution began. Morgan found a white-eyed male running around in one bottle. I am unaware of a single other example in the history of biology where one physical animal made such a difference. So in honor of that animal, there shall now be a brief, silly spiel to try to wake you up for the remaining 17 minutes. Most people would argue this is one of the 2 or 3 greatest opening sentences in the history of the English language. It is a truth universally acknowledged that a single man in possession of a good fortune must be in want of a wife. Which, of course, is the opening line from Pride and Prejudice, of course, by Jane Austen. And it, of course, describes the appearance of Colin Firth, which has much inspired many residents of that town to all sorts of things. Now, I'd like to put to you that the sentence I'm about to show you was written by Thomas Hunt Morgan, is no less lovely both in its rhythm and implication. And that's Thomas Hunt Morgan, and this is the opening sentence of his paper describing what he found from studies of that fly. He goes, in a pedigree culture of Drosophila, which had been running for nearly a year through a considerable number of generations, a male appeared with white eyes. The normal flies have brilliant red eyes. As I'm about to show you, that white-eyed male, which appeared in a pedigree culture, had quite an effect on the local population. And so, to make it easier for me to explain his life to you, I'm going to call him Darcy. So this is a wild-type fruit fly, and that's Darcy. He had a remarkable marital history in Thomas Hunt Morgan's lab. So I'm going to show you the crosses that Morgan did and the interpretation of the findings. So this Darcy shows up. There it is, this white-eyed male. The first thing you do when you're a geneticist is you cross it. That's what geneticists do. They run crosses. So you take Darcy and you cross Darcy to a wild-type red-eyed female fly, and all the progeny are red-eyed. Yawn, go get a cup of coffee. This is, of course, Mendel central. It's just like yellow times green times equals yawn. Fair enough. When, however, Morgan crossed the offspring to each other, so he essentially selfed the F1. Incidentally, from whose papers do you think he learned that trick to self the F1 after having made a hybrid? Mendel. Remember I told you that Mendel established the experimental paradigm for genetics for the rest of eternity? He did. What he came up with is the way we still do things. You cross the Darcy's daughters and sons to each other, you get red-eyed males, red-eyed females, white-eyed males, and no white-eyed females. Ugh. Oh, is this a male-only trait? Morgan was so puzzled by this finding that he then decided to cross Darcy to his own daughters. And Drosophila males live long enough to allow you to do that experiment. This is one of the main reasons Drosophila is such a marvelous model system. And he found in the offspring of that cross red and white-eyed males in equal proportion and red and white-eyed females in equal proportion. So whiteness of eyes is not a male-only trait. We've just made white-eyed females. That leaves us with the puzzling question of the outcome of this cross. What's going on? And to Morgan's eternal credit, he thought back to work by his own student, Nettie Stevens, who first of all had a fantastic taste in hats. Second, much more seriously, discovered the X and the Y chromosome and discovered the relationship between genders, the sexes, and their sex chromosome complement. And this is Morgan's own beautiful drawings from his book about this. XX versus XY, females versus males. By the way, why was the X chromosome called the X chromosome as opposed to the something else chromosome? It's because the person who discovered it didn't know what it did. So by analogy with the X-Files or the X-Men, that person called it the mystery chromosome, or for short, the X chromosome. Morgan thought to work by his student about the distinctness between the two sexes with respect to their XX and XY-ness. And then he realized, in one of the most probably glorious light bulb moments in the history of science, that the reason for the peculiarity of behavior of eye color inheritance is the Drosophila cross has to do with the fact that the gene alleles of which explain the white versus red difference must lie on the X chromosome. This was the first ever realization that Mendel's factors, the genes, lie on Fleming's threads, the chromosomes. I wanted to show you first Morgan's own beautiful drawings from his book before diving into images from your textbook, which are very simple. I'm going to fly through this, pun, awful pun not intended, because it's unbelievably simple. Yes, the white gene, and it is called white as opposed to red, even though its normal function is to cause redness, is because in Drosophila genetics and in other genetic systems, there is a convention to name the gene after the recessive phenotype, after the loss of function phenotype. So the white gene, which makes red pigment, when mutated, causes eyes to become white. That's why it's called white and not red. Sorry. So Morgan realized that the white gene lies on the X chromosome, and therefore Darcy is hemizygous. And so the F1 generation of Darcy's daughters looks like this. They're all red-eyed. And then when you cross them to each other, this happens. And I'm just clicking through figure 13-8 in your textbook, which I know you've read. And you've studied this in multiple contexts already. So I'm going through this very quickly because it's actually very simple. I simply wanted to emphasize to you that this was the first example in the history of science when somebody focused on the concordance of the behavior of phenotypic differences that appear to follow quasi-Mendelian ratios. and the behavior of chromosomes to realize that genes, alleles of which cause trait differences, actually lie in chromosomes. How are we to explain this remarkable cross? Just really quickly, there is a 13.8 has two examples of crosses that Morgan did. In this example, you take the white-eyed female, which you've just obtained, you cross it to a red-eyed wild-type male, you get the following ratio of progeny, and so on and so on and so on. What am I expecting you to know about this? What I'm expecting you to please do is simply sit down and follow the extremely simple logic of figure 13-8. What were the crosses done? What phenotypes were observed? How are the phenotypes observed? Explicable in terms of the white gene and its alleles lying on the X chromosome. Do you need to memorize this? No. Would I like you to understand this? Yes. Is it very simple? Of course it is. This allows me to spend a little bit of time talking about this. This, of course, is the most famous example of, quote, sex-linked inheritance, unquote, in the human genome. This is a disorder of blood clotting, or hemophilia. It was known as the royal disease in the late 19th, early 20th century. Most prominently, of course, it affected that boy. That is Prince Alexis. We know that he inherited the allele of the factor IX gene, blood clotting factor IX, which lies on the X chromosome, from his great-great-great-grandmother, the Queen Victoria. Now, this is Grigory Rasputin. Some of you may be under the misimpression that he founded a music store on Telegraph. He did not. He was a charlatan who convinced Prince Alexis's mother, who had severe blood clotting disorder and thus needed to be biked around because every time he would take a step there would be bleeding and it's really painful. He convinced his mother that he can cure the boy's hemophilia by laying his hands on him. Nothing could be further from the truth, of course. Unfortunately, the boy's father, Nicholas, listened to Rasputin's advice, and that changed world history. Remarkably and stunningly, 2 years ago, they exhumed the royal remains, and they actually got some DNA out of Alexis's remains, and they genotyped him. And we now know that he had factor IX deficiency. He had a splice mutation at the beginning of exon 4 of the factor IX gene of the X chromosome. So it's kind of surreal to have this voice of history preserved and speak to us back from 1917. Those of you who got here early were regaled with this rather remarkable sight. This is a pretty remarkable gene. And the reason it's rocking there for you. It is called SRY. This stands for sex reversal on the Y. And it has this rather colorful name because, folks, and I swear I'm not making this up, it is the foundational gene of manhood. The default pathway of human development and all mammalian development is, in fact, female. Very early after conception in embryos that are XY, that gene, which lies on the Y chromosome, is expresses a transcription factor, this protein you saw bound to the DNA, and it changes expression of genes in the urogenital ridge to transform the ovary into the testis. It's been experimentally shown that if you make point mutations, such as this, or all of that, in the SRY gene, then you have an XY individual who is phenotypically female. So is sex reversed. It is kind of surreal to me that, manness, fondness for sports center, is completely eliminatable by one point mutation. And you will say to me, what is the cognate female gene? There isn't. Femaleness is everywhere in the genome. It's the default pathway. Take out your clickers, please. All right. I'd like you to please spot the error. 1, 2, 3, 4, 5. 1, or A, DNA replication takes place during this step. 2, or B, the pairing of homologs that takes place underlies Mendel's first law. 3, or C, the fact that the blue homolog goes to one cell and the red homolog goes to the other explains Mendel's second law. D, or 4, the sisters separate at that step. E, this is a haploid cell. Okay, don't rush. I'll give you, let's see, I'll give you at least 90 seconds. Go. As always, feel free to ask your neighbors, Google the question, call your parents, better yet text your parents. All right, let's take another 45 seconds and then move on to the next slide. All right, 5, 4, 3, speak one or forever hold your peace. Okay, fine, done. Let's see what you said. Good job, very nice. Very nice. I am glad this happened because 194 of you, I mean, nice try, need to revisit Mendel's second law. And part of the utility of this response system is that I can sort of monitor how many of you understand certain core principles. I'm going to start walking you through major implications of Mendel's second law for mapping by linkage. And you can't study it without understanding Mendel's second law well. So let me take 60 seconds to give you a preview of what I'm hopeful you would understand, and then I'll let you go. And by the way, I have office hours today. Okay, first of all, let me quit this. Go away. Go away. All right, 60 more seconds, folks. I promise this will be worth it. The two most famous undergraduates in the history of mankind, I think. When you work in somebody's lab and you wash dishes and you feel sad that nobody appreciates you, think to Calvin Bridges and Alfred Sturtevant. They were undergrads in Morgan's lab. He couldn't afford regular research assistants, so he paid them as bottle washers. And Bridges was so poor, he couldn't afford clothes, so he couldn't go to parties. So he just stayed in lab all day, washing bottles and making medium. And one day he walked up to Morgan and said, "Professor Morgan, this fly has eyes that are a different color than the other flies." And Morgan said, "You have just been promoted to mutant hunter." It's a true story. And these two went on to found, together with Morgan, modern genetics. So the specific part of Mendel's second law I need you to understand is this, and with that I'm going to let you go. The example I'm walking you through has a serious problem. Bridges discovered purple-eyed flies. Until then, Morgan only had white-eyed flies. Sturtevant discovered flies with rudimentary wings. Oh, vestigial. You cross double homozygous dominant with double homozygous recessive. All the offspring look like the wild type. You test cross the double heterozygote to a double mutant fly, and Mendel's second law says you should see a 1 to 1 to 1 to 1 ratio of 4 phenotypic classes in equal proportions. Instead, you see a massive overrepresentation of the phenotypic classes that characterized grandma and grandpa, respectively. Before you come to Friday's lecture, study Mendel's second law in meiotic dynamics. until you understand why Mendel's second law cannot be used to explain these numbers. So I have office hours today. From 10 to 11 in 125 Li Ka-shing.